And today our speaker will be Associate Professor J. Andrew Jones. And Professor Jones has a PhD in Chemical and Biological Engineering from Rensselaer Polytech. And he conducts research on engineering microbes for high-value chemical production. And he's responsible for the first heterologous system for psilocybin production, as far as I'm aware. He's currently associate professor in the Department of Chemical, Paper, and Biomedical Engineering at Miami University. Without further introduction from me, I'm going to hand over to Dr. Jones.
Thank you for the introduction. Hopefully everybody online can hear me okay. I'm excited to come today and tell you a little bit about some work that I've done in my lab to try to make psychedelics, but making them in a platform that can enable really efficient and high production, efficient production of these microbes and bacteria. Great. First off, a little bit about Miami University. We are also a state university in Ohio, but we're a little bit different than Ohio State, right? We have an undergraduate focus. In the College of Engineering at Miami, we do not have a PhD program. So my lab is predominantly filled with undergraduates, and I have like one PhD student from a different department. But in my department, we have about 16 faculty members, a very small population of master's students, and a number of about 300 undergraduates. We pride ourselves in our undergraduate education, right? That's kind of shown in kind of US News World Report rankings. These are the latest ones that just came out, saying that we're the 11th best undergraduate teaching university, number 30 in kind of engineering education in programs without doctorate degrees, right? So that's something we pride ourselves in. That's kind of where we see ourselves making that impact. And that's kind of the perspective I'm going to bring this to you. Much of the work that you see here, the papers were first authored by undergraduate students in my lab, which is kind of crazy to think about, right? That you have undergraduate students that in many cases can't legally drink alcohol yet, but they're going to be biosynthesizing psychedelic drugs. So that's something that I think is kind of cool and kind of a neat introduction. I want to first start out a little bit about my field. I realize that a lot of the people that are maybe tuning in today are don't know what metabolic engineering is, or kind of the way that I attack problems. So I want to tell you a little study about a paper that came out, I think in 2015, where they engineered a yeast to produce hydrocodone, which is kind of a known pain-killing drug, right? Where it's traditionally made through poppies, right, and extracted, but they synthesize it using Saccharomyces cerevisiae, right? So I'm going to tell you that story to give you kind of a perspective on the field, right? And then I'm going to kind of, in parallel, talk about some of my work, right after that. I'm going to talk about some of my work to use E. coli to produce psilocybin, right? But then that's some of the work that we published in kind of, I guess, late 2019, and then we're going to expand on that in the last few years to talk about other derivatives, right? Whether that's DMT or 5-methoxy-DMT or other intermediate products in the psilocybin pathway. We're interested in a lot of those, and I'll talk about some of the future work. I'll talk about some unpublished data that we don't have yet, and I'll give you the state of the art of where my lab's at and where it's heading in the future. So, diving first into metabolic engineering, right? So this is a representative of at least part of the metabolic map of, let's just call it E. coli. I don't know what organism I'm for, but pretty much all organisms are equally complex. There's lots of enzyme catalyzed reactions, a lot of spontaneous reactions. There's cycles for regeneration of cofactors and precursors. They're very involved, right? So let's just imagine for a second that we want to engineer this system, right? We can imagine that changes on one side of the map can influence things on other sides of the map, right? So let's zoom in. Let's say there's this pathway that's being expressed inside an organism of your choice, right? It starts at glucose, right, at the top center, right? And let's say if we go down and to the right, we land at butanol, right? Let's say that's our target product, right? Pretty valid product for somebody that works in the metabolic engineering space. It can be mixed in with many fuels. It could be made pretty efficiently. But if I wanted to make more butanol from glucose, right, using synthetic biology and enzyme overexpressions, I could, or synthetic biology and kind of all types of recombinant technology, I can knock out competing pathways, right? I can funnel flux towards butanol. I can knock out things that might be influencing or pulling flux away, right? I can overexpress the pathway towards the end product, right? To make a lot of butanol, and it, in theory, should be that simple, right? If you increase the width of the road heading towards butanol and you shut down all the side streets, everybody's going to go the direction you want them to go. But there's more at play there, right? We might need to think about balances, right? Thinking about electrons or reducing equivalent balances. How does overexpressing of these enzymes mess with the NAD plus, or NADH balance within the cell, right? There's energy at play, right? So you can't make energy from nothing, right? And glycolysis can generate some energy. How do we consider that? Energy balances, does that matter? Does that change how pathways function and perform? And it certainly does, right? But we can also think about key metabolites. That acetyl-CoA is not only used to make butanol, it's not only just an intermediate in this, but it's used in a plethora of other pieces of metabolism. It's one of those interconnected nodes, right? So if we pull a large flux of that away, it's going to cause kind of health ramifications elsewhere in the brains, right? And we can go on and on and on and on with different applications that could be confusing and complicating factors that make it really difficult to understand how this pathway is going to perform, right? And it's oftentimes going to make things not work the way that you want it to, right? And this is the world that I live in, right? I try to think about ways to balance and use cellular resources in a way that kind of can enable the pathways that I want without kind of wrecking everything else that nature's kind of evolved over millennia. So jumping into this hydrocodone case study that I talked about previously, right? This great paper came out in Science in, I think, 2015. A group out at Stanford produced opioids. Ultimately, they made hydrocodone and yeast. And they got a lot of press, right? They were able to use baker's yeast to brew these narcotics. There was this review that came out in Nature that talked about this work, saying that we might need to regulate these home-brewed opioids, right? They really spelled it out. They said, in principle, anyone with this yeast strain and basic skills in fermentation will be able to grow this morphine-producing yeast using a homebrew kit for beer making, right? Like, they spelled it out, and of course, like, the popular science articles go crazy saying that, like, homebrewed opioids are a reality. The people on Reddit were really interested in investing in this lucrative opportunity, right? This made kind of large press. But as a scientist, we should ignore a lot of that, right? And we should go back to the science, and we should dig into the paper and try to figure out how much they made, right? And if you really dig in, it wasn't even enough to make it to a figure, right? It actually just made it into a section of the results. And they said they make 0.3 micrograms per liter of hydrocodone, right? And so I don't know how to measure that. Like, it seems like a small amount. But if we think about 5 milligrams as a typical dose, right? Small dose of this opioid to relieve pain for just a couple hours, and we convert some units, we're going to find that you're going to need almost 17,000 liters of culture to make enough product for one dose. And that's if you ignore all the downstream processing, right? Purifying 5 milligrams out of 17,000 liters is fundamentally impossible, right? So yeah, it's a really cool pathway. They pieced together a very long biosynthetic pathway and did some cool science, stuff that I'm excited about academically. But the practicality of it is kind of limited, right? So the takeaway message here is that you can get it to work, but proof of principle is not where you can stop, right? You have to keep going. You have to make it to a commercial product before you can really make that impact, right? So my argument here is that it's a good start, but they really have a lot of need for some further genetic engineering. This work has been commercialized. They're working on building out some of that. I'm not sure about the progress over the last 7 or 8 years. But keep this in mind when you see some of the results from some of the work I'm doing and how maybe I've tried to go a little beyond that proof of principle study. So diving into some of the work from my lab, I'm going to start by talking about mental health. And I apologize for the online. I have to find a way to get this stuff. All right. Either way, let's talk about mental health, right? I think we can all agree that mental health is prevalent, right? Worldwide, about 1 in 8 people have a diagnosable mental health condition, right? But if we look more locally to the US, that number grows to about 1 in 4, right? Those are your coworkers, your students, your colleagues, your friends, right? Your family members, right? Everybody is impacted by mental health, right? It's expensive. Those that live with a major mental health condition, right, live about 10 years shorter than those who don't, right? And so that's equivalent to a smoking addiction, right? Which is kind of crazy to think about, that like this thing that's so prevalent is just as bad for you from a health perspective as smoking, which I think is globally understood as not great for your health, right? But it's also expensive in terms of money, right? In the US alone, we spend about $190 billion is the cost for treating mental health. And this is a holistic number that includes kind of lost wages and time away from work and like potential loss. And, but that's a value that's roughly equivalent to the entire GDP of Greece, right? And that's in the US alone. That's not even looking to the global numbers, what that could be. So we're talking really large amounts of money involved with this. But the good thing to know is that mental health is treatable, right? We have pretty good antidepressants and anti-anxiety medications out there that work for a lot of people. But despite spending almost $200 billion, incurring $200 billion of cost for this, about a quarter of people that seek treatment have unmet mental health care needs, right? The current available technologies do not work for them. And that's because they work through a very narrow range of mechanisms of action, right? And finding new strategies to treat these diseases with new mechanisms of action is something that I'm working hard in my lab to try to develop drugs that can kind of fit that bill, right? But there is hope, right? The FDA in 2018 and 2019 gave a breakthrough designation, which is a fast track status that they give to 2 psilocybin trials that are currently underway. I guess some of them may be completed at this point. But we are working. There are dozens of clinical trials that are studying psychedelics because psychedelics have been shown time and time again to have some positive impacts here, right? It's still yet to be fully clinically proven and an approved drug, right? But those are ongoing, and we are really hoping that those have that potential in the future. And this motivates the need for some of my work to be able to produce these compounds in an efficient and effective way, to make them available for further study, to make them available for potential patients. So this is something maybe to this group that I don't necessarily have to spend a lot of time on, but this is a really cool plot, right? On the y-axis, you have dependent potential. You think about that as how addictive are these compounds, right? And on the x-axis, it's the active dose over lethal dose. Think about it as a safety profile, right? So if you look in the top right corner, the things that are very addictive and not very safe, you see the classic things you would expect, right? The morphine, heroin, cocaine, things that we would probably all agree are not great. But if you look just removed from that, you start to see things like nicotine, caffeine, and alcohol, right? Things that we know probably aren't in making our lives last longer, but we wouldn't think they would be that closely related or that connected to some of these known dangers, right? I always thought it was funny that I couldn't draw a circle between caffeine and alcohol without hitting roofies, but that's just me. But if you look down in the kind of far left, right, you start to see the psychoactive compounds, right? Things like THC, LSD, psilocybin, right? These are things that have been documented to have high potency, right? Really a high safety threshold, right? As well as low addictive properties. And so that's something that I think really motivates that these compounds might be not as dangerous as maybe people sold them to be in the 60s, 70s, and 80s. So let me get into more specifics into kind of my work. When we think about the biosynthesis pathway, we start by leveraging the promiscuity of the native tryptophan synthase, TRPV, right? We use E. coli for most of our work, so this is just naturally expressed. We're not overexpressing this enzyme. We just got really lucky, right? If we feed a 4-hydroxyindole substrate, so indole is a natural metabolite, right? We feed in a slightly modified hydroxy version. That's naturally condensed by the promiscuous action of TRPV into this non-natural amino acid 4-hydroxytryptophan, right? That then proceeds through previously published pathways, right? To first decarboxylate into 4-hydroxytryptamine, phosphorylate into norbaeocystin, and then there's 2 methylation steps catalyzed by Psy-M that create baeocystin, and finally our target end product, psilocybin. This is a slightly different biosynthesis pathway than what you see in the mushroom, right? The natural kind of psychedelic psilocybin mushroom. But it is one that bypasses a P450 step, right? The PSI-H that's naturally in that pathway that doesn't express well in E. coli, and I'll show you some of our data that we have gotten that to work later in the talk, right? But this process that was developed kind of in my lab is DNA encoded and autocatalytic. Means that we've engineered this DNA system, right, that it runs on its own, right? We put these cells into media with glucose, they grow and divide and create more copies of these cell factories that can produce this compound. It's a one-pot synthesis, right? If you compare this to a competing technology such as the chemical synthetic, right, there's many different steps and purification strategies that results in a really low yield, right? We have a really low synthesis cost, right? We can synthesize this product for a fraction of the cost of the chemical synthetic derivatives, which is one of the reasons why our pathway, or our production technique is competitive in the marketplace for production of these drugs. We also get more efficient. We produce more of this product as we've scaled up to larger and larger scales, and we've gotten better and better control systems for the fermentations. Those things have worked incredibly well. I'll tell you a little bit of that story in a minute. But I think we can all agree that it's way cooler than organic chemistry for farming, sorry. But I think a biosynthetic approach is the future for the production of these drugs, right? It takes away a lot of the variables and adds in a lot of known kind of standardizations that we've developed over years, learning how to ferment these compounds or ferment other products, right? Many pharmaceuticals are made this way. We've been making fermented foods and beverages such as beer at very large volumes, very efficiently for a long time, right? And this leverages a lot of that knowledge to be able to produce these compounds in that form. Okay. So when we started this project, I had a couple undergrads in the lab the summer after I started at Miami. And I was like, I got this harebrained idea building off of some work that other people have published. And I want to see if we can get this to work in E. coli, right? So we took the 3-gene pathway, we fed that precursor compound, and we just used kind of a standard starting point, right? We had a really high expression plasmid. We built it in a pseudo-operon configuration, the details of which are not particularly important for this conversation. But at that time, we had no standard. Like, we had no idea where this is going to elute, right? We hadn't purchased kind of the standard materials, but we had some idea. And when we ran our controls, we found like one tiny little blip on the HPLC chromatogram that like looked like the shoulder of some noise. And we were like, I don't know if that's it or not. So we were able to partner with a collaborator in chemistry at Miami, and we were able to look at a mass spec. We didn't have a mass spec in my lab at the time. And we were able to find a primary ion that matched the mass that we were looking for with psilocybin, and this was an Orbitrap mass spec, so it had really high res data. But then we're also to get the second mass, right? We were able to fragment that main ion and see what it breaks into, and it broke into exactly the sizes that we expected, right, for this particular compound, right? It gave us kind of the classic kind of predicted breakpoints, and we got the sizes. So we're pretty confident that we had made what we wanted to, right? This is one of those days when you sit back and it's like, I tried to make this go viral at one point. A Groundhog Day in science, like the Groundhog Day is when you like see more winter or spring's coming, right? And so like that's the day in the lab when you see like, this is a publication that's going to come, right? Or maybe not. But it really makes you sit back and say, whoa, like we just, for the first time in the history of the world, a bacterium has made psilocybin, right? And I hope that the Slot Lab proves me wrong and finds a bacteria that can do this at some point. But as far as I know, that was the first bacteria in the world that had ever potentially gone on a trip. Who knows? But again, we'd only made about 2 milligrams per liter of psilocybin, right? Still better than the opioid case that got published in Science, but we won't go there because the first thing we did is we said, now that we have a proof of concept, we have that proof of principle, we can start to scale up. We can start to optimize and tweak the pathway, tweak the conditions, right? And so we first started with some promoter and genetic optimization, right? So we screened about 400 different variants that we build in kind of a library-based system, and then kind of pick individual colonies and screen them and check it a few times before we go and sequence. Just by changing the promoter out in front of this pathway, right, and changing the orientation of promoters and terminators within the pathway, we were able to find a near 30-fold improvement in production. Right now we're up to 60 milligrams per liter. By tweaking the process parameters, thinking about how much substrate do we feed at the beginning, right? And how much of some supplementary amino acids do we put into the media? Can we boost the production, right? At that point, we're up to now 150 milligrams per liter. And then we jumped into kind of a scaled up story, right? But this kind of glosses over a lot of the details, right? But I want you to see, in this next step, I'm gonna walk through those same stages of optimization that I take to a lot of the problems in my lab, right? And I want you to see what that looks like from a metabolite plot, right? And so in this plot, on the x-axis, I have the metabolites in my biosynthetic psilocybin pathway, right? Starting on the left with the first metabolites going in order all the way to our final end product. So when we started, our original proof of concept looked like this, right? We had fed 4-hydroxyindole, most of that had gotten consumed, right? And been converted into the 4-hydroxytryptophan at pretty high efficiency. But it got stuck there, right? The next steps in the pathway, the true enzymes related to the psilocybin biosynthesis were kind of limiting, right? The next step is the Psi-D, the decarboxylation step, right? But through some genetic optimization, right? That promoter and transcriptional unit optimization, right? We're able to see kind of a shift, right? We're able to see more of that 4-hydroxytryptophan being consumed, and that's effectively pushing flux all the way towards that end product where that begins to build up, right? And then when we tweak some of the fermentation conditions, optimizing medium supplements, induction timing, things like that, we're able to see even a further push that gets us to that kind of final, where our predominant metabolite in the pathway that's remaining is the psilocybin. We can't make much more than what we made here because we had already consumed all of our previous metabolites. This is in batch phase, we're not feeding things in the intermittent stages, right? And so this is like the best story, right, that I can tell. Like, this is when science works, right? What I didn't tell you is like the 400,000 things we tried that didn't work in between, right? But from there, we went into our bench scale bioreactors. In my lab, we have 4 of these we can run in parallel. And we were able to then optimize parameters that we couldn't do in batch scale. We can now control pH, right? We can control the dissolved oxygen level. We can monitor and vary temperatures. Particularly, we can feed our 4-hydroxyindoles concentration in very slowly and keep it at low levels to reduce toxicity profiles. And all of that together was able to yield final titers that were over 1 gram per liter, right? We're getting 500, 600x higher than where we started, right? And we were to do all this in about 6 months, right? When science works, it works well, right? And so this is kind of what that data looks like that we published in that paper. If you look at your green line there in the middle, that's our psilocybin titer, and you can see that ramps up to over 1 gram per liter within kind of a 2.5-3 day period. Right? So that kind of tracks with our cell growth. So you can see that it happens very quickly, right? On time scales that don't necessarily, that can't be competed with in the traditional synthetic chemistry or growing kind of mushrooms on that side of things, right? It's also incredibly scalable, right? We went from 25 mil shake flasks to 2-liter bioreactors in kind of one swoop. And the information we have here has now been scaled up to 300-liter reactors with an industry partner. Okay? And so we published this paper in 2019. And then when this paper was published is when my life changed forever. Right? That's when I started to get lots of people reaching out for discussion. Right? I never thought that organizations that I've valued, NPR, Scientific American, would be interested in writing an article about some of the stuff I've done, right? I never expected High Times and Vice News to come talk to me about my research, right? Like maybe something else at some point. And not everybody got it right? The scientists engineered bacteria that poops magic mushrooms, like, might be a cool research idea, but did not do that. I never talked to Paul, but I would like to if he ever sees me do one of these talks. But I think this is where I want to take a second and kind of have a note to graduate students, right? And I encourage you to not think that the project ends when you submit it for publication, right? Self-promoting your work and reaching out to publicists and organizations that would like to summarize your work and write it in a way that non-scientific audiences can get a flavor and an idea of what you're doing and the impact of your work will greatly increase the impact of your work. It will make your work more valuable, right? And so now when I publish papers, I try to make sure that they get at least some type of non-peer-reviewed press, right? But that might be that I have to write that article and send it to someone to see if they want to publish it, right? But if you read any of these publications, right, you can reach out to the authors that write things in an area that kind of interests you, right? They're always interested in new stories. I find that they really do want to talk to you about some of the cool science you're doing, right? There's lots of value in that, and I encourage everyone to not see publication as the end of the story, but rather just the beginning, right? That's the thing that gives it validity, right? But it doesn't give it the impact, right? But then I went to Reddit, and Reddit loves this stuff, in case anybody hasn't like dug around. So we got some people wondering if we're going to hit the black market. These people thinks it's exciting times for the home brewing of systems, and then this guy went on a rant and kind of told everybody exactly how to do it. But he ended in the thing that I'm still waiting on, but he said, plus in a week or two of running even a small reactor at 1 gram per liter would put you at an extreme surplus of product and money, considering a dose of psilocybin is $35. Still waiting on the extreme surplus of money, but maybe that's coming. I'm still waiting. So Angry Zay is out there. Only one in the market. But based on this, it made me start to think, like, if somebody got my strain, right, which by the way is not regulated by the DEA, I don't hand it out, but if they were to get it, is there a potential that they could produce psilocybin by themselves? Or does it need some special sauce? Do you need to be trained in this area to be able to figure this out? Getting back to that review on the opioids that came earlier. So I asked the question, can psilocybin be homebrewed like beer? Like, what's holding us back, right? So what I did is I took a group of like green freshmen, right? And I said, I got this idea. I'm gonna give you a sous vide out of somebody's kitchen, some water to like help keep the temperature control. We're gonna do some aquarium pumps to add in some oxygen. We're just gonna set those bad boys on stir plates, right? We're not gonna sterilize things. We're just gonna give it a little rinse with some ethanol, toss it all together and see what happens, right? And I didn't really think it would work because there's a lot of difference between this and the bioreactor I was producing things in earlier. But it turns out you make hundreds of milligrams per liter of psilocybin using these engineered strains, right, in batch culture, in a system that looks like this, right? And we changed a few variables and we found some things mattered, some things didn't. But the end story is you can make quantities of this drug that would be biologically active in an environment that you could do in your garage, right? Which was kind of crazy. And we wrote this paper not to be a how-to guide on how to brew your own drugs, right? We didn't put the details in there that are required to do that. But we wrote it as a way to look at the regulatory kind of landscape. What things need to be done to both enable science, but also protect people from just making recombinant organisms and brewing their own drugs, right? There's definitely some health side effects that need to be considered there. So you can check out that paper if you want to see a little bit more about kind of some ideas on better regulatory kind of practices and some problems with the current practices. But to kick it all off, we were able to produce these compounds at a cost that was about 2% of what the equivalent street value of mushrooms, right, number of doses would be, right? So it's also like way cheaper to do it this way than it is to grow the mushrooms on ground, which is kind of the value of those mushrooms. So then I was interested in investigating kind of other natural tryptamines. I'm getting a bunch of chats, so I'll make sure that I can hear questions. And so the other products that are made in psilocybin, the other parts of the pathway, right? So the precursors that I introduced earlier, novacysteine, baecysteine, I was interested in what they can do. Like, can we produce these as well? Maybe they also have some therapeutic efficacy for something, right? And organocin is the step beyond psilocybin if we add an extra methyl group onto that. And I had this hypothesis that these molecules are more than just intermediate products, but they interact when the whole mushroom species is taken on consumption, and they do something, right? I think they have some value that's more than just a way to get to psilocybin. And that different mushrooms act on people differently because they have different ratios of these active components in them, right? But the problem at the time, there was no commercial availability of these compounds. Today, there's a limited commercial availability for some of these. They are still come at really high prices that limit a lot of the studies that people want to do. So I set out to produce these myself, right, to be able to make them available to potential collaborators that might want to study them, might want to learn more, kind of help understand some of the mechanisms of action of some of these compounds. Okay? So the psilocybin story that I've kind of already given you, I want to do a parallel with when I tried to do it again and make norvastatin. Okay? So we started with the psilocybin pathway, which is the full psi, D, K, and M, right? To make norvastatin, you just take away the M, right? So a very similar pathway construction, right? We're able to do a very similar genetic optimization approach where we modified and screened hundreds of pathway variants, and we're able to identify kind of genetically superior, genetically elite mutants that were really good at producing these compounds. It turns out they worked even better for this compound because there's much less burden to making norvastatin than psilocybin, mainly because you don't have the methyltransferases that have a SAM cycle or a methyl donor that is pretty complicated to regenerate. And then in the end, once we scaled up in our bioreactors, instead of 1 gram per liter, we have to get about 1.5 grams per liter of this norvastatin compound. Right? The interesting thing here is that when we go to purify it, they have very different chemical properties, right? Which makes it have to take a very different chemical purification process, right? Which is something that I don't know has been talked about in the literature, right? Until we started to present some of this work. I then partnered, now that I had some purified compounds, I partnered with a collaborator in psychology at Miami that works in the animal behavioral space. And he had a few kind of models that he had built for not necessarily psychedelics at the time, but he established them once we started this collaboration. And the first I'm going to introduce is this head twitch response, right? Which is the idea is that rodents, when they're going through what we perceive as a psychedelic experience, right, they shake their head more frequently than they do without, right? And there's a specific intensity, a specific frequency that they shake that head at. And so I've got a little video here, and there's a couple different head twitches. So that's one, and then that's another one, right? So it's just that little fast twitch, right? And so the way that we kind of measure this is we do a small surgery on these rats, and we put a magnet on their head, like we fix that in with dental epoxy, and then we put those rats, after being dosed with the drug, inside a bucket that's lined with a metal coil, and that electric field, the magnetic field inside the electric coil, gives out a signal that we can then process using some signal processing software to quantify when these head twitches are occurring and count them. They're then verified by kind of video on top of that. This is a process that was developed in mice originally, and we kind of changed it into a rat-based method. And so here's some results from the 4 compounds that we're interested in. Interestingly, for psilocybin, we got the results that have been published before, right? We see a dose-dependent response, at least up to 1 mg per kg, and then it kind of dips off at that high dose, right? That's pretty standard for a lot of the pharmacology in the space, right? But bassist and bassist and urination didn't see any head twitch response, right? Which suggests that those are going to be non-hallucinogenic, right? Which initially I was like, bummer, that's unfortunate, right? Because I thought that this hallucinogenic component was the important part, right? But what I'm thinking now is that might be beneficial, right? Because one of the things that's holding back psilocybin from becoming a therapeutic treatment, being approved by the FDA for, let's say, depression, one of the things that's holding it back is this side effect that you have to go on a 6-hour trip, right? When you want to take these drugs, and it's inconvenient. It makes it more expensive because you have to be monitored during that time, and you have to kind of control that environment a lot more, right? So if we could find a compound that could kind of break apart the psychedelic part from the mental health treatment part, or the actions of those kind of biologically, that could be valuable, right? So I said that maybe this isn't bad news. Maybe one of these drugs is something that is of interest, right? So we, in Matt's lab, they continued to test some of these, but we went to some different testing, right? So we then did a sucrose preference testing, right? Which is the idea that rodents, just like humans, like sweet solutions, right? So you prefer the sweet. So a non-depressed animal is going to take the sweet water bottle every time over the non-sweet water bottle. But when you have a depressed rodent, they kind of lose that preference. They're like, whatever, water is water. And you see that kind of, and then so when you give a depressed animal a antidepressant, right, they kind of transition back. They revert back to liking the sweet solution more. That's kind of the premise behind this study, right? This is one, so fluoxetine is our positive control here, and it is the active ingredient in Prozac, right, or known SSRI. And it doesn't work in this case, actually. It actually shows no reversal of that. It looks just like the kind of vehicle control, the negative control. And that's well documented that all of these models that we're using, they don't work for everything. Right? So the idea is you want to do multiple things and you want to kind of see what the full picture looks like in the end, right? Prozac, fluoxetine, works really well for a lot of people to help treat depression. But when you look at psilocybin, norvastatin here, and then we had a combined one, which we won't go into the details of that here, but they show kind of that reversal. They show a statistically significant improvement, right? We didn't see anything for the other two, the Biosynthesis and the organizing. So that I'm not showing type of argument here. But then we went further and we did what's called a forced swim testing. So the idea is you put these animals, they're swimming in a bucket of water they can't escape from. And the question is, how long do they try to escape, right? Even though they realize they can't get out, right? And a depressed rat will be immobile for a longer period of time. Right? So the idea is that if you are not depressed, you're going to struggle and try to get out for longer. So we see here in our vehicle control, we see a pretty high immobility duration, right? But we see fluoxetine, right, Prozac, psilocybin, and norvastatin all seeing a decreased immobility. Yeah. This is after treatment. So the question was, are these results under the influence of the compound, or is it like an after effect? So there was a treatment dose. I don't have all the details in front of me, but I want to say they got 2 doses. One was like the day before, and one was a few hours before, something like that. There's a standardized protocol for this, for antidepressant testing of these compounds. I believe these were healthy animals too, as well. They were not depressed. So these are potentially muted responses compared to what you would see in a kind of chronic stress. So this gives us some promise. It says that, particularly in the case of norvastatin, something that is non-hallucinogenic, shows no head-twitch response, is showing some interesting positive impacts in the animal behavioral space that says that maybe these compounds could be good for antidepressant, right? So it could be that golden ticket. We're still working on that answer, but it's something that's pretty exciting. We're getting ready to publish some of this data really soon. We then started to kind of think, what other derivatives could we make in this kind of pathway? Check the time, make sure we're good. So this is some work that came out this summer, where an undergraduate, Jessica Flower in my lab, and one of my research associates worked together to develop this technology, where we took our existing psilocybin production strain, right, the one on the left, and we added in a new strain that was expressing only the P450 hydroxylase, right, and the corresponding reductase partner, to be able to, one, enable the de novo production. Can we make psilocybin from glucose, right, not having to feed that 4-hydroxyindole substrate? But by doing that, we then open up the ability to feed in other substrates that aren't 4-hydroxy and make a variety of different psilocybin derivatives, again, with the hope that we can make enough of these and start to screen some of these compounds in some of these animal models to figure out if we can find something that might be a better drug than psilocybin, right? Something that's more easily synthesized, or something that's more easily taken or controlled in a blinded study. Right? So this was the structure we did. It's a proof of principle. You wouldn't want to scale up a system that has kind of a 2-strain co-culture. That doesn't quite make sense. But it was a way for us to kind of screen these things incredibly rapidly using an already optimized psilocybin production strain, so we didn't have to worry about high efficiency and those kind of other steps, and allowed us to focus on that P450 step. Right? And so the way this thing worked is we bought a library of, I think, 40 plus, I think we're over 50 now, different indole derivatives, right, that have different functional groups at different positions on that compound. And we fed those indole derivatives to the wild type strains to see how promiscuous that tryptophan synthase, that first step, was going to be. It's kind of natally made in cells. And then for the ones that worked, the ones that we saw some promiscuity and saw the production of that tryptophan, non-natural amino acid, we then used them in a strain that had Psi-D, right? So then we had both of the first 2 steps, and we tried to see, can they make the tryptamine? So we were able to measure the promiscuity of that first step in the enzyme, in the pathway, right? And then a lot of those didn't work really well, right? So then we changed how that Psi-D was expressed, and we tried to optimize that single gene in the pathway, and we're able to see that improve a little bit more, right? And that optimization piece kind of is where it really expanded, because we took 40 plus indole derivatives and we screened them across 8 or 9 different strains or promoter variants to have different expression levels, right? And surprisingly, we saw a wide range of different activities, right? So in this particular case, right, this is just an example, in the H9 promoter in our system worked 4 times better than our kind of native T7 promoter that we probably started with, right? So the way this looks over the library of kind of all 40 is this first plot here is our TRPV. So this is our tryptophan synthase, and the numbers you see in the boxes are percent yields, right, under a standard set of screening conditions that we picked to kind of be fair across the whole board. And you can see that out of the, I think there's 45 here that are represented, you can see, I don't know, 38 of them or so are showing some activity. So we're seeing pretty wide substrate promiscuity, but they don't all work really well. There's lots of numbers here that are 1%, 5% yield, right? But we see a lot of hundreds as well, right? The 7 position seems to be very flexible for that tryptophan synthase, right? Where the 5 position is not so flexible, right? Or the 6 position. Then taking that forward, using the T7 promoter for our Psi-D, kind of our first initial screen, we were a little disappointed. Some of them worked really well. Things that we got lucky with in that first story, right? The 4-hydroxy, you see how the 4-hydroxy indole there in that little blocks, 92%, right? Had a really high yield. We didn't engineer it that way. That was the one we started with. We got lucky that this whole psilocybin process worked early on. If we would have ended up with one of these less than ones, we probably would have had a very different kind of trajectory on some of this science. But when we optimize the Psi-D expression, we're able to see a lot more green pop up. We're able to push that to near 100% yield. And it's a little bit misleading because it's 100% yield of the available tryptophan, right? So if the tryptophan synthase didn't work very well, the Psi-D didn't have to convert all that much, right? But we didn't have a library of tryptophans to be able to feed as well, right? It's prohibitively expensive for us to handle. But interestingly, we've built off of this in the last few months, I guess, where we've started to fill in a lot of the gaps here, right? We've started to get different decarboxylases, different tryptophan synthases, right? That have been engineered to have different promiscuities. And all of a sudden, some of the ones that are zeros or single digits are starting to become 100%, right? And my goal for the lab is to get 50, right? I want to have abilities to produce high amounts of 50 different tryptamines, right? So that I effectively could make 50 different psilocybins in there. Not quite there yet, we're getting there. But this just gets us to the tryptamines. We then, I'm gonna skip this slide for now, because we then extended that pathway past the Psi-D to include the Psi-K and Psi-M to fill out that whole psilocybin pathway. And we're able to see de novo psilocybin, but more excitingly, we were able to find 13 different psilocybin derivatives that have been produced. I'm showing the data here for kind of methyl psilocybin, right? You can see the giant peak that pops up right in the middle, has the right molecular weight. And we're able to see this time and time again. We're able to track all the intermediates, right? And by looking specifically at all the intermediates in this whole pathway for this large library of indole derivatives that we start with, we can start to understand the promiscuity of not only the first couple of enzymes, but we can look at the entire pathway of enzymes. And we can tell you that the P450 is very limited, right? It has a very narrow range. And everything that got through the P450 went all the way to the end product, right? We didn't see anything stop. We didn't see Psi-M be limiting at all, right? Psi-M has to have the phosphate group out there, right? But it doesn't need to have, but it's very insensitive to a lot of variations of kind of mainly halogens around the ring of that indole structure. So we were able to get to some really cool evidence here. It was published this summer in Biotechnology and Bioengineering. So I encourage you to take this out. It's some of the work in my career that I'm the most proud of. I then had a master's student in my lab who came to me and says, I want to make DMT. And I was like, all right, we'll try it, right? The pathway for DMT was not necessarily published, right? There were some rumblings online of what some of these pathways might look like. But we started and we built from what we knew, right? We knew that we had active tryptophan synthases, right? We're not only interested in DMT, but we're also interested in bufotenin, which is 5-hydroxy-DMT, as well as the 5-methoxy-DMT. And we know we had the promiscuity of that first step, that tryptophan synthase, to be able to make those derivative products, right? We also knew PSI-D was very promiscuous, right? And it could take it up, and we can make those tryptamine derivatives as well. But it's that methyltransferase that we didn't know, right? The SIM, the psilocybin methyltransferase, has to have that 4-position phosphate group to be active. It doesn't act on things that don't have that 4-position phosphate group. There's been some discussion that maybe a 6-position phosphate could go, but the phosphate's important regardless. DMT doesn't have that, so how can we do this? So we're able to find an enzyme that's expressed in humans. We use the human variant of it, which is rare when you're working with bacteria. Hopefully that doesn't work. And we're able to see kind of activity for it, right? A little background I forgot to say. DMT is the compound that comes in ayahuasca, which is this drink that's natively brewed, or tea that's brewed in kind of South American religious and cultural studies. 5-methoxy-DMT and bufotenin were found in this guy, Enceladus arboris, the Colorado river toad, something like the glands behind the ears there. They excrete those compounds. It's kind of a venom. But we were able to achieve, like after we went to a bioreactor and did the scale up, we were able to achieve 75 milligrams per liter of this DMT, but we were also able to make a pretty even higher amounts of NMT, which is the immediate precursor, right? And then measurable amounts of TMT, which is kind of the quaternary amine, right? There's some interesting kind of details in there, but we were able to piece this pathway together that naturally exists in organisms without using any of the DNA components that actually came from those organisms, right? Which is kind of interesting. As we learn about the promiscuity of these enzymes, we can learn how to piece them together in new ways to make new drugs and new pathways. This is something that's pretty exciting. Shifting to kind of the focus of my lab in the future, right? I really get excited about making new compounds, right? If I can make some new compounds, I'm hoping that someone will help me find a use for it, right? Or if similarly, if somebody's like, I wish you could make this compound, right? That's motivation for me to kind of try. I'm really focused on the education of it, right? Engineering, or like working with my undergraduate students, graduate students, to teach them how to be good scientists and how to go out and solve big problems that have no answer currently, right? It's something that gets me excited, right? And so I'm looking for how to do that with a cool project. Right? So I want to expand the number and diversity of these chemical entities that I'm making, right? Derivatives on psilocybin. With some idea that maybe this idea of psychedelics without the trip is something that at least gets me excited, gives me, I see a kind of potential path forward for that. So that's kind of the direction I'm looking. I'm also putting a lot of effort into scale up, right? Both better understanding the upstream production side of things, right? Can we get that 1 gram per liter to 10 grams per liter? Because that makes a lot of problems a lot easier. Then also on the downstream side, can we make a scalable downstream production method that could in theory make kilograms of products, so that if we do find something of interest, right, there could be a potential to run clinical studies with those compounds, right? But in the end, I really want to make testable amounts of compound, right, that I could provide to a growing list of collaborators that they could use in their models to study something, right? Learn something about these compounds to help improve our understanding of this kind of powerful class of molecules, right? It's something that's really exciting to me. And then that leads to the impact piece, where I want to expand the number of my collaborators, right? I want to talk to people in medicine, neuroscience, and biochemistry, and animal behavioral people, right? I want to get people from all areas that are interested in studying different aspects of these compounds, right? I'm good at learning how to produce these compounds and thinking about the kind of biochemical processes, right? But I want people that care about the neurochemistry and the neuroscience and the neuroplasticity of these compounds and understanding how the structure-function relationships build into that space. And I want to try to facilitate that any way that I can. That's where I see my lab fitting in the future. And it's one of the reasons I'm here, is to be able to look for people that might be interested in some of this and kind of share a central theme, so I can learn about what compounds they might be interested in and see how we can have some synergies. With that, I didn't do this alone. I have a huge lab of really talented people that work incredibly hard, right? And this is just a snapshot of them. I got a whole list of people that aren't in the pictures. And all of them have helped out in one way or another. Many of them, the majority of them, are undergraduates, and I have a growing list of collaborators across the country that I'm hoping to kind of expand that list, and I want to get these compounds to as many people as I can, facilitate their study. But with that, I'm very thankful to be invited. I hope you took something away from it. I hope you enjoyed the talk. And I think we have some questions.